more efficiently collected. So it's really a marvel of engineering that we're learning from and has been now applied to numbers of experimental products, including the dashboard protecting plastic over your instruments in your car, eyeglasses, military optics. They will also help increase both the efficiency of solar cells and their ability to collect light throughout the day, as well as reducing the cost of production of those solar cells. So if our goals are met, we will have contributed a little piece to that big pie in the sky of being able to get free, limitless energy from sunlight.
Creative Microsystems uses nature as their guide. Another example, the Lab on a Chip, miniaturizes medical diagnostic equipment by mimicking a butterfly's tongue.
We're reaching for the day where we're doing stuff pretty much the same way that nature has done in evolving organisms to be exquisite sensors. One of the interesting devices that we've been working on for one of our clients for a few years is an artificial tongue. It has a very thin film that is built as a flat, almost like a diving board that hangs out in space. Now keep in mind the sizes of this. It's about 10 nanometers thick and about 10 nanometers wide and about 100 microns long. So it's a very, very thin structure that will interact with only one kind of molecule, and it curls. And we can see that curl by bouncing a beam of light off of it. So there are a number of applications for that in chemical sensing and in biological detection and in medical applications. That device, in its general sense, is referred to as a lab on a chip, where we take entire laboratories worth of equipment and put that onto a single piece of silicon or glass or plastic. The possibility of making structures that are much smaller than a human cell, for example, concerns that are very much on the minds of people that are working in those fields or in the field of nanotechnology. Notion that this is dangerous is certainly something that's being explored very actively by people in the field that are policing themselves and getting government support to do so, trying to head off any potential concerns that the public may have about building structures that could affect their cells or affect their well-being.
In addition to these fascinating examples in biomimicry, Creative Microsystems has years of expertise in the creation of holograms. They are currently breaking ground in the fabrication of integrated circuits using holography.
Very exciting project for us. It's been ongoing now for quite a few years, about 10 years. Originally supported as a Phase 0 grant from Vermont EPSCoR. And at the time, we didn't know where we were going to go with it, but we knew that there would be a way to use a hologram to support, or in some cases actually replace, what is now done in semiconductor fab using big lenses and very complicated and expensive machines called steppers. The laser is the heart of all of this. It produces pure light where all of the waves of the light basically stand still at their relative positions. So we can mix the beams together. When they mix, they combine in a predictable manner. And because they are nanometer dimensions themselves, a lot of the light we work with is in the area of 2 to 250 nanometers of wavelength. So when you mix two elements of the same wave with the same wavelength, then we can get what are called constructive interference recordings. A good example would be taking a look at a surface of a pond or a puddle or a swimming pool. If you throw an object in, you get a lot of ripples. Those ripples will travel out until they hit an edge. Well, if at one instant in time you were to capture a picture of that edge, you could then play that back in time and reconstruct the object that made the ripples. And that's exactly what holography is. It's a picture of the ripples modified by something in space, illuminated by light. And that permanent record that is in the surface of the material, that becomes the structure. We can use a very powerful laser that has a very large amount of energy in each photon. Those photons get to be what we call expensive photons. In a standard mask, where the light is going through little holes where you have some of the light getting through and some of it not getting through, inherently you're wasting a lot of those expensive photons. Our holographic masks completely utilize the light that's put on them. And with a single flash of light, lets us do things like pattern integrated circuit materials in single pulses of light. We don't wait around for the kind of chemistry to occur in the old-fashioned way. We actually go in there and blow holes where we need a hole, or vaporize the material where we want to have a line, and cut out a large amount of the time and also a lot of the resources that are necessary, like water, making a more green industry. I think the calculation is somewhere in the order of 100 gallons of water is needed to produce one integrated circuit chip. Some of our processes use no water whatsoever.
Bill's nanoscale optic design could revolutionize the fabrication of integrated circuits. Back at UVM, we'll return to the fabrication technique of nanomaterials using the well-established process of electroplating. Frederick Sanzos is integrating these into the construction of composites.
You can use a large amount of nanomaterials and mix it with a structural material to make a composite material. But the composite will be really strengthened because those tiny particles or tiny wires inside the material are very strong. Right now, this research is still in its infancy, so we are still working in the lab to make those devices.
So what we got here is an AAO template, and we're going to take this, and how it's used to grow wires is it's going to act as a mold. It's a thin membrane, and it's got tons of tiny nanoscopic holes in it, and each hole acts as a mold for an individual nanowire. When we grow the wires, we'd take this template and fully seal it with electrical tape. This is the template that we're positioning above our Watts solution for electrodeposition. And here's the nickel electrode. We're going to lower them both in. Once they're in position, we can induce a current between the two electrodes to create the electrodeposition process. Nickel inside the Watts solution is going to be forced to go to the back of the template, as seen on here, and grow out. So each hole is an individual mold for each individual wire.
This is one of ZAC membrane that has been electrodeposited. We simply place the membrane like this and cut this membrane in two halves. And I'm going to focus and zoom in. So this is the thickness of the membrane, which contains tiny pores. And as you can see on this image, and this indicates that we have grown metallic nanowires. And what this vial contains is simply billions of nickel nanowires in an ethanol solvent. One way to know that we have exactly nickel nanowires, I should see some wires attracted by the magnets. And as you can see here at the bottom of the vials, actually large bundles of those nanowires. And to show you the scale at which we are working, I place a human hair next to a bundle of nanowires. Here's the hair. Okay, this represents 100 micron. So you can see that's about half of it is 50 micron diameter. And next to this wire, to this hair, I have a wire. So I'm going to zoom in. Here's an example of a nickel nanowire. One challenge here is to make enough nanomaterials to build a house or a car. So that's one challenge. But I would say within 5 to 10 years, this challenge should be overcome.
When nanofibers are integrated into composites, a stronger material results for recreational equipment, automobiles, and construction material. Over at the chemistry lab, we find another exciting application of nanotechnology, the increased efficacy of cancer drugs.
My research here involves inorganic chemistry. We work with a lot of solid materials, and in particular porous materials. There's lots of different things you can imagine porous materials doing. And so we're doing some tests in collaboration with the medical school that is involved in fighting cancer. Whether we can enhance the efficacy of a drug simply by not changing anything about the drug itself, just changing about how you get it into a cell. One advantage that these materials have, extra volume and extra surface area inside. So per gram, in other words, of material, a dense material that didn't have any pores, you couldn't really, say you're looking to load it with a particular drug, you couldn't really put that much of the drug in it because there's not that much surface area per gram of material. But if our materials have 100 or 1,000 times the surface area, that means potentially we can load 100 to 1,000 times the drug inside the particle. The material that we use, the delivery device, if you call it that, which is an inorganic particle, that's something that we make all the time in lab. What we do to make our nanoparticles, we'd start with a chemical that we'd call a monomer, so a single chemical on its own, just dissolved in solution. And then we add it to another solution that help those molecules react with each other. And then the pores in the material are created by surfactants that are present in the material as well. The critical aspect of the surfactant is that it has a positive charge. The surfactant is a template around which the reaction happens, and it polymerizes around that template. So it's a positive charge of the surfactant and a negative charge of the silica. The shapes of the pores that we get out when we're done with everything after the furnace are directly related to the type of surfactant that we have. And at the end of that process, what we end up with are these beads. We take it over to the medical school. So we pack that into a pipette tip, a very small amount, micrograms is what it is. And we mix it in a very small amount of liquid that is the same liquid that's used to grow cells. And then that mixture is then added to a cell culture that's in a mini petri dish. We can show at least a 30 to 50-fold improvement in the number of cancer cells that are killed if we use our particles to deliver the drug. If our research is any indication, nanotechnology has distinct promise for improving the efficiency of drug transfer, and that will lead to definitely new treatments and better treatments for cancer. It's nice to do things in a lab and think about them, but when you're really working with people, I think that's, shows that things are really coming to fruition. It's chemistry that's in life,
Whether it's chemists working from the bottom up, finding new ways to enhance our health, or engineers fabricating nanomachines from the top down to fuel our progress, it's clear that there's incredible potential in nanotechnology. At the same time, our scientists reminded us that there could be unintended consequences when manipulating at the molecular level. On both fronts, Vermont scientists are certainly leading this work. It's been exciting to learn about this research and to expand our understanding of its impact on our lives. I'm Amy Seidel. Join me next time as we explore more emerging science. Wow.